Yes, this is my first time holding this meeting on my own, so I hope everything goes well.
Yeah.
I hope that this really goes well, because I really, when I heard what the topic was, I had that kind of not seeing the trees from the forest. This is my first time holding this meeting on my own, so I hope everything goes well. I hope close that and hope
I'm going to say mute something or close your window. Yeah, okay. Yeah,
no, it happened. Anyhow, yeah, it was kind of a not seeing the trees from the forest kind of moment, because I've never thought of antimicrobial resistance and COVID kind of being in the same sentence.
Oh, really?
Okay. It just makes total sense, and it's so obvious, and it really is something that should be thought of as really important. But I don't know, I've always kind of thought about it as in vaccines and strains and what's going to happen with the virus itself, and is there going to be enough like vaccination immunity, like are enough people going to be vaccinated and everything. And it's just, and then I thought, oh
yeah,
it's so obvious. It's such a biologist way of thinking that just never crossed my mind. It just kind of opened up such a new perception for me personally. So I'm really looking forward to this. Like, I think it's going to be super interesting. So really, like, we're super grateful and thank you for joining us.
No problem. And so what's your background? Are you a molecular biologist then, or?
Yeah, I just finished my undergraduate studies and I'm just doing my master's in genomics.
Okay. Oh, cool. Okay.
Yeah.
And then what are you going to do?
And then I'm searching for a PhD at the moment. I'm applying for them. So very stressful, but it's going to be okay.
Yeah. And is there much research on AMR in the University of Belgrade? That's where you're based as well, is it?
So hold on a second. Yeah, excuse me. Somebody from my house decided that now was the appropriate time to come in. Sorry, that's the reality from, doing it from home.
Exactly. Yeah. I'm borrowing an office for today. So,
yeah, so let me just start letting people in. Okay, so I'm going to make a couple of people co-hosts and we're going to start in a couple of minutes. Just so that if anybody is a bit late, they can join as well. Yeah. Okay, so I've made a couple of you co-hosts now. So in case my internet goes away, as it tends to, you can continue the meeting instead of me. Okay. So we'll just wait a couple of minutes and then we'll start letting the rest of them in. So are you okay? Like how are things in London in respects to the whole thing? I have some friends in England and they say that it's kind of cut loose at this point, that No restrictions.
Very few restrictions. I just tried to share my screen and it said host disabled participant screen sharing. So if you could just make me
Okay.
make me co-host as well, maybe. Does that
Yep. Let's try that. Can you try it now?
Yes, I can do it now. Yes.
Okay. Troubleshooted one thing.
Yes. You want to admit those in the waiting room as well that I can see?
Sorry?
I can see there's people waiting in the waiting room. You're admitting them.
Yeah, that's the rest of the people. So people from the actual society I've let in, but people just, I don't know how to say, regular people. I haven't. So we're just gonna go to admit all.
And will you share the questions at the end or something like that? Or?
Yeah.
Okay.
So we're just gonna, okay. So kind of the regular way we have it is you can just have the lecture in the format you want it, and then all of the questions could be at the end, or they can type it into the chat whilst it's happening.
Okay.
Okay, so I think we've waited long enough. So I'd like to thank you all for coming so much. So this is a lecture that I'm really personally excited by, and this is a lecture in the organization of the Student Society for Molecular Biology. And this lecture is part of our Antimicrobial Awareness Week, so that we've dedicated a whole week to antimicrobial awareness. And Professor Gwen Knight has graciously decided to agree to host a lecture for us. And the title of the lecture is Antimicrobial Resistance and COVID-19, its intersections and implications, which to me personally is a very exciting topic and something that I personally haven't thought about and probably should have. So Professor Gwen Knight is currently an associate professor at the London School of Hygiene and Tropical Medicine, and she obviously has a fellowship, and her fellowship tends to explore the dynamics of antimicrobial resistance from 3 different angles. So she builds models that explore the resistance in sort of fitness and resistance of antimicrobial resistance, and then the how different places have different levels of resistance, and how we use this heterogeneity to build models for better intervention design, and how we can use that to control antimicrobial resistance. And obviously she currently works in COVID-19 responses. I feel like everybody is, and she's focused on how SARS-CoV-2 spreads in hospital environments. So I'm gonna stop talking now. So I'll let the professor do the talking, and we can have questions at the end, or you can type them into the chat. Okay, the floor is yours.
Thank you. Thanks. Hello, everybody. I presume you can see my screen. Can you see my screen?
Yep, we can see the presentation.
Great. Okay, now you should see it full screen, right? Yes?
Mm-hmm. But
Okay,
we can see the note slides on the side.
Oh, can you? How do I make it like that, right? No?
Nope, no, we can't.
Oh, how do you not see the notes? Oh, that's annoying. Sorry, this is a new, how's that? No,
Yeah,
no notes? Yeah,
Now
cool.
it's,
Sorry, I'm on someone else's computer. Use a quieter office. Okay, cool. Hello, everybody. Thank you for the introduction. Yeah, my name is Gwen Knight, and I'm an associate professor at the London School of Hygiene and Tropical Medicine, also deputy director of the AMR Centre at the school. And I work within the Centre for Mathematical Modelling of Infectious Diseases because that's really my focus. I use mathematical models to explore the dynamics of antimicrobial resistance, as was mentioned in the introduction, and also as mentioned in the introduction, as a mathematical model of infectious diseases. Obviously, I can't avoid thinking about or working on the spread of SARS-CoV-2, and so that's what led me to think a bit more about how my two research fields collide and how COVID-19 is going to impact on antimicrobial resistance in the future. So just to give you a bit of an outline of what I'll talk about in the next 20 to 25 minutes, I sort of thought I'd start with a bit of background in terms of what is mathematical modelling, and then very briefly about what is antimicrobial resistance. I think you're probably all experts on that, especially if you're here for World Antimicrobial Awareness Week, but just a little insight into that. And then bring those together to give you some examples of how I use mathematical modelling to address the problem of antimicrobial resistance. And then move on to think about how COVID-19 will interact with AMR, and can and should we model the impact of COVID-19 on AMR, and what do we know, what might we think is going to happen with antimicrobial resistance or AMR in the future. Okay, so that's an outline of my talk. I'm happy to take clarification questions during the talk, but it's probably easy to just hold questions to the end, maybe put them in the chat and then you won't forget them. Okay, but I probably won't look at the chat during the talk. Okay, so I'll show you this lovely picture here of someone sneezing. And what you can see is the spread of droplets over time from A to F from this individual sneezing. And I show this because when we think about infectious diseases and antimicrobial resistance and COVID-19, we're thinking immediately about the clinical implications, or we're thinking about the biology, we're thinking about the genome sequencing, we're thinking about what is this? Is this a virus? Is this a bacteria? Sort of fundamental questions. But I would also argue that there are a lot of quantitative questions that we could and should be asking. So, for example, here, how far do those droplets spread after a sneeze? How big are the droplets? How much virus or how many pathogens are in each of those droplets? And then that translates immediately into things like infectious dose. So how many of those droplets need to land on a second individual for that second individual to be infected? And what is the size of that infectious dose? Is it 1,000 bacteria? Is it 10,000 bacteria, et cetera, et cetera? So from something that initially seems to be a very clinical or biological topic, infectious diseases, there are many different quantitative or mathematical questions that I think need to be answered, and they immediately come to mind. And that's really where mathematical modelling comes in. It allows us to use mathematics to change these questions into equations or into mathematical quantifications, such as the R number, so this number that we're probably all aware of due to COVID-19, so the number of secondary cases caused by one infectious individual. And that's really a combination of many of these different factors. So how likely is it that someone is to come into contact with somebody who's not infected? How likely is it when they come into contact, transmission occurs? And we can break these all down into mathematical questions and then build up mathematical relationships that we can analyze using mathematics. So I really see mathematical modeling as like another tool that we can use to answer questions about the spread of different infectious diseases, or for me in particular, the spread of antimicrobial resistance. And there it's like another piece in the research toolkit, very much like we use animal models. It's an accepted but unfortunate part of modern science that we use animal models as sort of an off-the-shelf tool to represent, for example, the immune system. So the way that the drug or the bacteria causes an infection in an animal model, we can use inference from that to say something about how it might happen in a human. And mathematical models are a similar thing. We can build up in a computer a representation or a model of how we think the world works, how we think a disease spreads. And we can use this to ask questions about the different types of interventions that we might put in, or different hypotheses about how a disease actually spreads in a certain subpopulation, for example. And so models describe our beliefs about how the world functions. And in mathematical modeling, we translate those beliefs into the language of mathematics. So we all have models in our head of how a disease spreads or how an immune system works. And writing them down, quantifying that exact relationship, and actually just writing it down in black and white, I believe A interacts with B, more quickly than B interacts with C. Already that's making a mathematical model, and we're doing that automatically all the time. And by putting it down in black and white in mathematics, then we have a very powerful tool to answer our research questions. The key thing to say, obviously, is that mathematical models have to be a simplification of reality. We can't include all the different factors. And so what we have to do when we build mathematical models is realize that our model is going to be wrong. It's not going to be an exact replication of reality. So we've got to be alert to what is importantly wrong. What did we leave out? What did we include? What is the impact of those emissions on our results? But it doesn't remove that tool. You know, we still use mouse models as a replication of human, even though they're not. We can use mathematical models to tell us something about our system, even though we know that it's not exact representation, right? So why would you build a mathematical model? So I hope I've sort of convinced you there that it has potential as a tool. One of the main reasons I see for building mathematical models, as someone who works on antimicrobial resistance, which is a hugely complicated and a hugely multidimensional field. We've got social scientists working in it, we've got clinicians working with it, we've got molecular biologists working with it. By building a mathematical model, you write down in very clear, mathematical sentences, but very clear statements and sentences what you believe is happening in your system. And when you do that, it becomes almost like a campfire. It becomes a focal point around which people can debate and argue and bring their data and challenge your hypothesis. And when they do that, they can learn more about the system. So I'd say that it's really what I see as one of the main reasons to use mathematical modeling, is to get a better understanding of how diseases spread and transmit, or how bacteria or how viruses spread and transmit. You build your underlying structure, you write it down in mathematical equations, you compare the output of that to the data. Am I capturing what we see in reality? And then if you're not, then you can adapt the model and build it around in this iterative process. And in that process, you're learning something about the key components of your system. You can also then use this model as a tool. So then you can say, well, at the moment, we think this many infections are going to happen over the next 6 months. What if we had a vaccine? What if we had a new drug? What if we had a new diagnostic? And you can test all those things out depending on the structure of your model to say what the impact of different interventions might be. And obviously, here I'm talking about mathematical models for infectious diseases. And so this is something we want to do. We want to know how to prevent the spread. We want to prevent disease and ultimately save lives. And so that really informs decision making. So which intervention should we put into place, or what would the impact of that intervention be, or what would happen if we didn't do anything? How many cases are we going to have in the future if we don't control the spread of this disease? To be very bland and boring about it and go down to what a definition is, I would say that a mathematical model is a description of a system using mathematical concepts and language. And this really gives us the advantage that then we have a precise and a concise language, which has well-defined rules for manipulations. And we have historical results of manipulations available, as well as a wealth now of computing power. So we're at the point now where we can really build quite complicated models of how the world works and test them in quite rigorous and structured ways against very complicated data. However, as I've mentioned, obviously it's always a simplification. There's also choices. You always have to make choices which equations to use, and there's always limitations. What do we know and what don't we know? What could we include and what couldn't we include, which make models a good tool, but also just one tool of many in our toolkit. And I would argue that, like any other research tool, has its advantages and disadvantages, but it is a great way of getting new insights into what can be very complicated systems. So I've talked a bit about mathematical modelling. Now I use mathematical modelling to answer questions around antimicrobial resistance. And I think for this audience, I don't have to define it, go back to basics. But what I'm talking about here is antimicrobial resistance, which is basically when pathogens such as bacteria, so that's mostly what I focus on is bacteria, they no longer respond to medicines, making infections harder to treat and actually increasing the risk of disease spread, illness and death. So this is the WHO definition of antimicrobial resistance. And so for me, it's something that's really important to work on, but it's also extremely fascinating because you've got the elements of evolution going on. You've got the fact that these bacteria are being exposed to medicines, and we're actually just applying a selective pressure. We're seeing evolution in action. And I believe that means that we can do something about that evolution, because a lot of it is human directed. And so that there's an opportunity there to intervene and to prevent antimicrobial resistance in the future. Now, I'm sure you're all aware of the data from the Jim O'Neill report from 2014, estimating that there's a huge number of deaths due to antimicrobial resistance, and that's set to increase. This is a sort of basic step to support why antimicrobial resistance is a really important area to work on. And as I'll talk about later on, we don't really know yet how this has been affected by COVID-19. All we know is that it will be affected. The other interesting thing about antimicrobial resistance is that it's not only a big problem, it's a global problem. So here it's an infographic from the Pew Trust, which shows that superbugs don't respect borders. And this is the spread of a gene, and New Delhi metamalases. Oh gosh, I don't think I've said that quite right. But anyway, a gene NDM1, which encodes for a certain type of resistance, and it was found to spread across the world. And we don't understand how it spreads. We don't understand the rate of spread, and we don't really know how to stop it spreading. And so there's, again, going back to mathematical modeling, this is like any other infectious disease. This is like the spread of SARS-CoV-2. How did it move? What are the selective pressures, and where could we intervene? What could we do about it? Because if we can understand what's happened historically, then we can go forward and prevent it happening again. I also really want to emphasize here that I think the answer to antimicrobial resistance is not simply the development of new antibiotics, although the pipeline is relatively dry, as shown here by the graphic of the number of new antibiotics that have been developed over the last 110 years or something. If we get a new antibiotic, resistance has been seen extremely rapidly. And so we really need to understand where AMR is coming from, because anytime then we get a new antibiotic, we need to be able to preserve that. And to preserve it, we really need to understand the evolution. We won't be able to develop new antibiotics. And even if we were able to develop new antibiotics relatively rapidly, it seems to be much more effective to preserve the ones we have and the new ones that we develop than to try and go back and get rid of resistance once it arises. So how do I use modelling for antimicrobial resistance? So I use modelling to answer policy relevant questions on the dynamics of AMR. So where does it come from? How is it moving? Where is it going to go? And I do this using both experimental and epidemiological data to really try to bring quantitative insights to how much, where, how many, how fast, in order to be able to inform interventions. And I can break this down because although I've said that antimicrobial resistance is complex, you can really think about it, or I think about it in three different dimensions. So antimicrobial resistance has to emerge from somewhere. So we have bacteria that are susceptible and bacteria that are resistant. And in some situations, the resistant population dominates, and others are susceptible. In some situations, the resistance genes from the resistant bug spread to the susceptible one. When did this happen, and how much and where? That's really the sort of focus of the emergence part. And then once resistant bacteria has emerged, how does it spread? So who does it spread to? How much does it spread? Where does it go? That's the sort of transmission aspect of it. And then the disease burden is sort of the practical epidemiology, public health side of it. What could we do about it? How is this trend going to change over time? And what kind of interventions should we be targeting based on our understanding of the emergence and transmission? So I'll talk now about a couple of different projects that I have in each of these areas, which use mathematical modelling and insights to better address the dynamics of antimicrobial resistance. So the first one of those focuses on emergence, and this is work that's ongoing with PhD students that I have that focus on the bacterium Staphylococcus aureus, in particular thinking about methicillin-resistant Staphylococcus aureus' resistant subpopulation of the bacteria, which is a common cause of hospital-associated infections. Indeed, it causes, I think it's the second biggest killer, or the bacterium that causes the second largest number of deaths due to resistant bacterium in Europe. At least that was from estimates a couple of years ago. So what we're interested in within MRSA is the rate at which resistance genes move. So how rapidly does a bacteria that's susceptible to an antibiotic gain resistance genes from a resistant bacteria? And so what we do is we use experimental data where we put together bacteria that are resistant to different types of antibiotics, and we measure how fast double resistant bacteria rise. So resistant bacteria that have both of the resistance genes from the parent strains. And the main way that resistance genes move in Staph aureus is by bacteriophage. And they basically, when they're a virus that kills bacteria, but they can accidentally mispackage and pick up bacterial DNA and transfer that between bacteria. And so what we do in our system is we add phage to this to try and mimic what happens in the human body and look at the rates of resistance gene transfer. And so on the left-hand side, you can see the sort of experimental setup, what we do in the lab. On the right-hand side, you can see the in silico model that we build, where we put mathematical equations behind all of these arrows, and then we try to fit the output from those mathematical models to the experimental data to inform understanding about how these bacteria and phage interact. And so some of those insights have been about what that model structure should look like and how we should capture the interaction between phage and bacteria over time, but also we're getting quantitative outputs. So we estimate that actually 1 in every 10 to the 8 new phage carries an AMR gene. So that means once in every 10 to the 8 interactions between phage bacteria, they accidentally pick up a piece of bacteria that encodes a resistance gene, and they transfer it to another bacteria. And so these kind of numbers, we're now thinking about how we can apply that to, for example, in a human, how often do resistant strains emerge within an infection, for example, a bloodstream infection. This also has implications for phage therapy, which, as we see the rise of antimicrobial resistance, we want to think about new ways that we can treat these infections. One way you could do that is using phage, because they're viruses that kill bacteria. However, there's sort of a double-edged sword, because we believe that, yes, they can kill bacteria, but they can also move resistance genes. And so we have to be very careful thinking about what might happen if we applied phage to a bacterial infection. We move now to thinking about transmission. The question we wanted to ask here is very different. So moving from Staphylococcus aureus to Mycobacterium tuberculosis. Pre-COVID, this was the #1 infectious disease globally. A multi-drug resistant TB is when the bacteria causing infection in an individual is resistant to at least the first 2 drugs for treatment, isoniazid and rifampicin. That's the definition of multi-drug resistant. And a latent infection is when someone is carrying this bacteria, but it's not actively causing symptomatic disease. But that individual is at high risk later on in life of developing TB disease. It's usually a lung infection. And we know that globally, around 23% of the world's population is latently infected with this bacteria, which is a huge number, but not all of them will go on to active TB disease. That's why you have this sort of iceberg effect in terms of how many individuals are carrying this bacterium. In this project here, what we wanted to do was to try to understand how many of those latent infections were resistant bacteria, because if a person is infected with a resistant bacteria, then in the future they could progress to disease with that resistant bacteria and go on to spread it to other individuals, but also be much harder to treat that disease in that individual. So what we did here was to build a cohort model, so to use mathematical modeling to track how infection might change over time using different forms of infection, different treatment rates, and try to understand the trajectory over time of infection with multi-drug resistant TB. What I'm showing you here is a snapshot of data of model outputs for Europe. So on the y-axis, you've got prevalence of infection. On the x-axis, you've got age in the different 5-year age bands. You can see this peaked effect. So what we actually found was that, unfortunately, it's likely that younger individuals are much more likely to be infected with drug-resistant TB because there's not this protective effect from a past infection. And we estimated that 3 in every 1,000 people globally are carrying latent MDR TB infection, and that prevalence is around twice as high in those younger than 15. So moving on now rapidly, I realise I'm giving very sort of brief snapshots of these examples. So if anyone wants me to talk a bit more about them, I can do so at the end. So moving on from this, to think about another angle of AMR, instead of thinking about where it comes from or how it's transmitted, we want to think about what we can do about treating it. Now, most bacterial infections, or most infections globally, when they're given, or most antibiotic therapy that's given, I'm trying to think what the denominator is there, whether it's infections or bacterial infection. But anyway, globally, when a patient is given antibiotics, usually they're given it empirically. And that means initially you give the patient antibiotic based on the symptoms that you can see, not on a diagnostic test that tells you what is actually causing those symptoms, whether it's bacteria or virus. So that's the definition of empiric. And in many settings globally, this empiric therapy, so what antibiotics you give a patient, is not based on knowledge of the resistance prevalence in that setting. So you might not want to give drug A because resistance to drug A is relatively high. And so in this project here, what we wanted to do was to say, well, can we do a bit better than that? Can we try and support, can we try and use global data sets that we have? Here we used a big one from Pfizer, which had just been released by the Wellcome Trust. Can we use global data on prevalence and resistance to better support countries and hospitals within countries to design better empiric antibiotic therapy? And here, the sort of switch that we made was, instead of reporting at a country level the level of resistance in a certain pathogen, so this country has 10% of E. coli isolates resistant to gentamicin, or some stuff like this, we switched the thinking to instead thinking about syndrome level resistance. And so we could say, and if we think about a syndrome like sepsis, what percentage of sepsis is due to E. coli versus another bacteria, so bacteria A and B, say they're 50-50. And then we want to know, in terms of treating a sepsis infection, what percentage of sepsis infections are resistant to drug X? And then we have to think about, if 50% are due to bacteria A, what percentage of them are resistant to drug X? Oh, none. Okay, so 50% of sepsis are susceptible. What if all the other bacteria are resistant to drug X? So that means that in terms of the sepsis syndrome, 50% of sepsis patients are infected with this bacteria, which is totally resistant to drug X. So we would then say, depending on your cutoff for treatment, so most places set that about 15 or 20% resistant, that if 50% of your isolates that cause sepsis are resistant to that drug, then you probably shouldn't use that drug to treat that syndrome. So there's a bit of a switch there, and we use modelling to combine these big data to then say at a country level what we would recommend in terms of first, second, and third line empiric therapy for that sepsis, for those different syndromes. And that's work that we've now got funding to work directly with a hospital in Zambia to see whether this can actually be used at the individual hospital level to better help them design empiric therapy. I'm sorry, and that's who we're working with. So that gives you just an example of some of the work that I've done and some of the work that's ongoing right across the spread of antimicrobial resistance, from emergence to transmission to disease burden. So all of this is happening obviously before 2020, when we had the outbreak of SARS-CoV-2 and now the ongoing pandemic. So I wanted to think about how these three different aspects of the spread of antimicrobial resistance were affected by COVID-19. Now here, I wonder if I can actually see it. Maybe I need to stop sharing, but I wonder if, oh no, I can see the participants. Can you put your hands up on the Zoom chat if you agree with the statement, due to COVID-19, there will be more infections with drug-resistant pathogens in 2020? Can people put their hands up? Yes, they can. Okay, I'm seeing you. Thank you very much. I can see the count going up. I've got 3 people out of 52, 5 people. Okay, I just wait until the numbers stop changing. Okay, so I'm asking you here to put your hand up if you think that because of COVID-19, there'll be more infections with drug-resistant pathogens in 2021. Oh, somebody put their hand up and then put it down again. Or maybe it's a time delay thing. No. Okay, so I think we're stable. Oh no, still going up. Okay, hang on a second then. Okay, so I've got 17 people. Someone's put a hand down. 17 or 18 going up again. Thumbs up. Oh, I don't know what a thumbs up means, but I'm going to count that as a hands up. Okay, so we're getting about 22, 23. Okay. So I'm going to say that it's about half of you on the call think there'll be more infections with drug-resistant pathogens in 2021. Okay, great. Okay, can everybody put their hands down? So then I'm going to assume that the other 30 individuals, or 20, 27 or whatever it was at the peak or something like this, 30 people, those 30 individuals think there will be fewer infections with drug-resistant pathogens in 2021. can I ask you to put your hand up if you think that it would be too complicated and you don't know whether it's going to go up or down? So I'm asking people to put their hands up if they think that it won't be clearly more or clearly less. I think I've got 2 historical hands there, but maybe those 2 historical hands. So you can put your hands up if you think due to COVID-19, there will be more infection of drug-resistant pathogen 21 in some places, fewer in others, but you're not really sure what it will be. So I've got 4 hands for this. So I've got about 23 people that think there'll be more. And then the rest of you think there'll be fewer, apart from 4 of you that think it will go up in some places, down in some places, but you're not really sure. Okay, 4 seems to be quite stable. Okay, great. Okay, I'm going to write that down. We'll come back to it. Come back to it later. Okay, so let me just close that. Okay, so lots of you think there'll be more infections with drug-resistant pathogens in 2021 due to COVID-19. So this is a question that I had in the middle of 2020. Obviously, as someone working on antimicrobial resistance in the middle of COVID-19, initially I started working with hospitals to see if I could do anything to support understanding of bed capacity needs, length of stay analysis, where is transmission happening. This is some of my initial work in that area. But then I started thinking more about, well, how is this actually going to impact my field of expertise, antimicrobial resistance? And so I started thinking, well, can we actually determine the impact of COVID-19 on AMR? Will we be able to answer this question? And then immediately I was thinking, well, what exactly is this question? Is it the impact on hospitals? Is it the impact on drug-resistant TB? Is it the impact on antibiotic usage? Is it the impact on the number of infections? Are we interested in just hospital settings? Are we interested in just high-income settings? And do we have any data? Do we have enough data is what I would ask as a mathematical modeler before starting on any topic. And for this, the question immediately was, do we have any data? And then once I started thinking about how we'd actually build this model, do we actually know how these 2 public health priorities interact? Some of the first sort of interesting work was starting to come out in 2020. So this is a really important piece of work. So this is a rapid living review, which I think has been, these living reviews, have been really important in COVID-19. And it seemed to come out relatively quickly that actually there was only a small percent of COVID-19 patients that had a secondary infection, so less than 20%. However, more than 65, so around 70 seems to be pretty steady now, of COVID-19 patients were being given antibiotics. So why are they being given antibiotics? Potentially not to treat an actual secondary bacterial infection. So we go back to this idea that most antibiotics are being given empirically to treat the symptoms, to treat the patient in front of you. And then when I tried to think about this impact, I started working with individuals across the AMR Centre at the London School to bring together all of our knowledge about how different ways diseases interact. And we ended up with some horrific mind map pictures. This is probably one of the nicest ones. We were trying to think about how COVID-19 is going to impact on all the different aspects of AMR emergence and spread. And so from this horribly complicated picture, we decided that we needed to write something about this and try to be a bit more systematic in thinking about how COVID-19 impacts on AMR. And so we really aimed to write something, which I think is the piece that that sort of triggered my invite for this talk today, to demonstrate that the impact, we really wanted to show that the impact of COVID-19 on AMR was complex, that it's not a simple relationship. And we wanted to use that to then propose mitigation strategies. Can we use that understanding to intervene? And then to actually consider the research landscape. So what needs to be done? What don't we know? And where should we go in terms of AMR and COVID going forward? And as I said, this is with many other individuals across the AMR Centre, and I was sort of spearheading it because I wanted to model it. So I wanted to build a model because I wanted to understand the underlying components, but it was just really complicated. So the first question we actually had was, instead of, if we're thinking about how COVID-19 impacts AMR, is there any interaction the other way? So is the way we consider COVID-19 and treatment of COVID-19 and patients with COVID-19, does AMR influence that at all? And the answer is yes, because many patients with COVID-19 get antibiotics due to their symptom profile. So they present at a hospital in the very serious stages of COVID-19, looking very much like a pneumonia patient. So you need to treat what you think, a clinician needs to treat what they see in front of them, which could be a very severe bacterial or viral infection. But obviously, if they're getting antibiotics, they think that it's a bacterial infection. And which antibiotic they choose, and hence the outcome or morbidity for that patient, is influenced by the AMR prevalence. And so AMR influences how we treat COVID-19 patients and which antibiotics they get given. And then the bigger question is, how will COVID-19 affect AMR? And this is where we took away those complicated mind maps and just tried to stick with some kind of table format within which we could include some examples of how we think COVID-19 will affect AMR. And that's what this infographic shows you here. So we split those 3 dimensions that I've shown you before: the emergence, the transmission and burden of AMR. And then we thought about the different components of our health systems and how COVID-19 is going to affect each of those. So we thought about antimicrobial use, how is that going to change? Infection prevention, what's going to happen there? And then the bigger sort of health system aspects to it. And when we did that, again, we had to break it down into these many different areas. And there's much more information in the paper if you want to see the references down the bottom there. But with all of these, we found that it was really not clear whether we could say that COVID-19 was going to increase AMR or decrease AMR. In fact, in many settings, it was kind of like, it's going to go both ways, and it's going to both increase the number of infections, but decrease the antibiotic exposure. Anyway, I'll just talk you through a few of those examples now. So in terms of antimicrobial use changes, obviously patients with COVID-19 exposed to a lot of antibiotics. It's a small part of the population, but many of them may have long-term sequelae, so they may end up with long-term more use. However, as we all know, there are many different global supply chain disruptions. They didn't just affect food and medicine, they also affected drug availability and medicine availability. So actually, if you've got no antibiotics in your setting, you're not implementing the selective pressure, which may cause resistance to increase. So actually, resistance may go down. And then in a broader aspect, there may be greater financial hardship due to COVID-19, which may increase the inability to pay for medical care, and therefore increased use of cheaper and potentially shorter duration counterfeit therapies, which have concentrations of antibiotics that may lead to resistance selection. So just a sort of snapshot of the different things, the ways that COVID-19 may affect antimicrobial use. We also then looked at infection prevention. So this sort of many of this goes to the transmission aspect of it. In high income settings, it's likely that you have fewer elective surgeries, so fewer healthcare associated infections potentially. And many of those infections would have been with resistant bacteria. So maybe we get a decrease in that. But then you might have more hand washing in the community, so a break in the transmission steps, and therefore fewer infections, and therefore fewer resistances, and also fewer antibiotic prescriptions or usage. And then there's the complexity of travel. So travel, we believe, in terms of international travel, that can often be a risk factor for spreading clones and infectious disease transmission. And that was stopped, so you might see fewer AMR infections. However, within a household, if everybody's home together, you're more likely to get saturation and spread of resistant pathogen to all members of a household. Finally, in terms of health system disruption, you've got this big problem that with diagnostic delay, you may see fewer individuals diagnosed, and therefore more transmission of the resistant bacteria. But those patients may also not be getting treated, so you may see less selection of the resistant subpopulation. And then, unfortunately, vaccination coverage is likely to be much lower in 2020 for many common diseases, and therefore we may see infections such as measles or flu leading to secondary bacterial infections. Actually, we didn't see that with flu, but future going forward, with things that should have been covered by routine, for example, childhood vaccinations, we may see complexity in terms of onward bacterial infections that lead to increased antibiotic use and increased resistance. That's a big snapshot. There's many different examples there, but I hope what I'm showing you is that actually we couldn't say conclusively one way or the other whether AMR was going to be influenced by which way AMR is going to be influenced by COVID-19. It will have different short, medium and long-term impacts, and will go in different directions depending on the setting that you're in. What was clear, though, was that AMR research would have been slowed by COVID-19. We now need support for AMR researchers to go back and use the networks that they developed and the tools that were developed for COVID-19 and apply them to AMR. And then we came up with multiple priorities, which I won't go through here. I'm happy to go through that in the questions if you're interested. But one of the most interesting things that came out that I thought was in terms of data and surveillance, we really aren't doing very well at in real time understanding the spread of antimicrobial resistance, and who should pay for that, I think is a really interesting question. So how has AMR changed? I think that even now we don't really know. We can't say clearly one way or the other. We're starting to get some data from the UK, for example. What we do know a little bit about is how antibiotic usage has changed. So again, in data from England, on the left-hand side here, you've got data in primary care, on the right-hand side in secondary care. Each of the different coloured lines is a different year, and the dotted line is the predicted, sorry, the dots, even the line of dots, is what was predicted for 2020 using standard sort of time series analysis. And you can see here that actually the total number of antibiotics used was smaller than expected across the whole population, but there's more broad spectrum antibiotics being used. So the black line is above the dots in the second figure. This might be, or this has been postulated to be, because fewer kids are getting infections because they had less, they were isolating at home effectively, so there's less transmission, and so fewer narrow spectrum antibiotics being given to kids. It's just a hypothesis, I think, at the moment. And then in terms of secondary care, you can see that actually there are fewer defined daily doses, so that's a sort of standardized unit for antibiotic prescribing, fewer than you would expect in 2020, but more per admission. So more per admission in secondary care and hospital settings, because admissions were probably down in many of these settings because of a lack of elective care. I should say this is England, so a high income setting. So actually in 2020, we may have seen a decrease in antibiotic usage. This is borne out by other work that was being done in North London, where you can see the predicted line in dots is higher than the actual number of antibiotics that were given. And also the same thing has been seen in the US, so decrease in outpatient antibiotic usage in 2020, which again may point to COVID-19 reducing actually the number of drug-resistant infections going forward. Now, if we want to try and model that, I was thinking about how you could do this in the UK, and the next couple of slides are really just my current thinking about things that we could do. What we could do is think about a pre-COVID-19 hospital and a during COVID-19 hospital. So how have things changed? Well, you've got lots of patients in hospitals getting antibiotics pre-COVID. That number has probably stayed the same, but now you've got this influx of COVID-19 patients, but you've got fewer elective surgeries and general care going on. And then the community, it seems like we've got fewer antibiotics being prescribed and a switch to more broad spectrum antibiotics. How will that influence AMR going forward? And how has that subpopulation of people in hospitals changed? And maybe the way to think about it is to focus on one bug or syndrome or drug and to try and understand that going forward. And so to answer the question about should we model the impact of COVID-19 AMR, I don't think we know the impact of COVID-19 on AMR. Should we model it? Again, I think we're just at the cusp of getting enough data to know that. We could look at what-if scenarios, and we could use those what-if scenarios to test against the data to see where AMR is going. We could think about looking specifically at pathogen that causes pneumonia, so similar to COVID-19, and see how that's been influenced. Could just focus on hospital patients. But then what's the point? What is the audience? So where are we with that? Anyway, that is my current thinking about what we should do about AMR and COVID and what we can do in terms of modelling. So I'm going to end there with just some conclusions. I hope that I've shown to you that mathematical modelling can systematically and logically reflect the models we have in our head of how systems work, and that is vitally important for something that's as complex as antimicrobial resistance, where you've got to think about emergence, you've got to think about transmission, and you've got to think about disease burden. And I would argue that developing new antibiotics is really just firefighting. Oh no, we need another intervention. We need to stop that infection. Well, let's step back a minute. Let's think about where it's coming from and use that to improve control. COVID-19 has definitely altered the AMR landscape. I think we're just starting to understand how. We don't know whether it will get better or worse, but we need to use this to understand AMR control and collect more data before we can jump in and do much modelling. And then I think we really need to stop as an AMR community now and think about what we can learn about COVID-19. For example, thinking about disease inequality, how does AMR spread differently in different populations? And how much AMR do we have, and where is it, and where it's concentrated, and what? And without that, we don't really know what we can do about it. So I'd like to say thanks to all the people that I've worked with on this, particular Quentin Leclerc, my PhD student, who seems to be involved in lots of the aspects of this, the UK MRC for my funding. I also like to say that I have a couple of PhD opportunities open. I've just opened at the moment with the MRC LID scheme at the school, one on empiric therapy for drug-resistant TB with Finn McQuaid, and one on resistance gene movement in MRSA with JD Lindsay. So if you're interested, either get in contact with me or have a look on the website and see. And those are my Twitter handle and my email address if you want to get in contact afterwards. But yes, I'm happy to take questions now. I think we've got about 10 minutes I can say around. So yes, thanks for listening.
Thank you so much for that lecture. It's been really interesting, and to me personally, I've always thought of the whole COVID-19 thing in strict biological terms. So obviously resistance would increase because we're treating something with antibiotics without kind of seeing where it fits into the real world. And I think that's why your research was so important, because it kind of put the whole thing into a real world perspective.
Yeah. But not everybody got COVID-19, right? Not the very minor proportion of the population that ended up in hospitals, and lots of those got antibiotics. But that's where the quantification of it comes in. Understanding the distribution of the patients in the population is really important.
Yeah, exactly. So thank you so much. And if anybody has any questions, you can just turn on your microphone or you can type it into the chat now.
Hello, I have one question. So first, thank you for the interesting talk. I didn't quite understand this experiment with phages and bacteria, resistance genes movement. So I guess it will depend very much on concentrations of bacteria and of phages in particular. So how do you even approximately match this to what happens in nature?
Say the last bit again. How do you even approximate? Oh, in a patient? In a patient?
How do you sort of match this with what happens in nature?
Nature, yeah. Yeah, so I mean, we're still at the amazingly, we're still at the early stages of understanding the interaction between phage and bacteria in terms of, as you say, the initial concentration. So what Quentin has been finding is that actually the interaction between the phage and bacteria is really dependent on those initial conditions. So we put in the same amount of bacteria every time, and then we put in different concentrations of phage, and we see that the phage and bacteria stabilize at different stationary phase levels. And actually that if you put in sufficient amount of bacteria, yes, it will shred the bacterial population. If you don't put in enough, it won't shred it. In fact, you get a sort of evolutionary, like a stable state where the bacteria and phage stabilize, which is not what's expected. It's sort of taken as read that if you add phage to your bacterial population, the bacteria is susceptible, that the phage will destroy that population. That's not what we see in our model. So already that's an insight that we gain. And then how does that relate to nature? Something that we really need to work on or understand is the number of phage and the distribution of the types of phage that we have, for example, in the human microbiome. I'm going to misquote the number, but I believe there are more phage in our body than human cells. I think that's definitely true because there's more bacteria, right? But there's a very, very large number of phage in our bacteria in our body. So we don't really understand how susceptible our bacterial populations are to that distribution of phage and how those populations are changing. So we're really at the early stages of starting with a very controlled environment. So if we have 10 to the 8 bacteria and 10 to the 4 phage, what happens? Changing that, understanding that, building a model for that, and then we can go forward to predict how you might use that to influence, for example, how many phage you should introduce into someone with a bloodstream infection, where you could measure the number of bacteria that they have in their blood and use that to tweak the phage therapy doses that we give them. Does that answer your question, Marco? I mean,
yes,
yeah.
Yes. Thank you very much. Thanks. So actually, I have another question, if there is time. I didn't quite understand when you're talking about the influence of COVID-19 on antibiotic resistance. So how you actually technically formulate this? Do you think about this when you say model? Do you think in terms of dynamical model, compartmental model, or you're thinking more in terms of like statistical learning, sort of machine learning problem where you would have, for example, antimicrobial resistance, and then you would have some predictors, right? For example, incidence of COVID-19, and you want to see whether it is statistically significant or not, or you, as I mentioned, really want to build a dynamical model with all the rates and all the things.
Yeah, that's a really good question. I think that's something that I didn't mention here, but obviously mathematical modeling could encompass statistical modeling. I'm more interested, or the models that I build tend to be more on the dynamic transmission side of it. So I'm thinking more about modeling individuals with and without bacteria, or with and without COVID-19 or SARS-CoV-2 infection. And then so building more of a dynamic transmission model to understand what happens over time and as those populations evolve and get given different antibiotics and transmission. So that would be more of a compartmental model, probably built with systems of ordinary differential equations. That's a lot of the approaches that I use. However, as you say, we're at the point where we might want to do statistical modeling first. So that's often what we do in mathematical modeling for infectious diseases, is that we start off by gathering the data, and then we do statistical analysis of it. So as you say, you could do some sort of regression modeling to look at the key drivers or the correlations between different drivers. So I think that's definitely something that we would do first of all, to see if there is any association to model, right? And then the strength of that association. So one thing I mentioned briefly there is something we should learn about with AMR, I think, is inequality. So there is some research that has looked into whether there are certain hotspots where there's higher levels of resistance than you'd expect, or more infections are due to resistant pathogens than in other places. Again, I'm sorry, I'm talking about England here. And it would be really interesting to see if those are also the hotspots of COVID-19, whether they're in the same age groups, for example. And then if you can understand that, then you know what you need to build in your mathematical model. So for example, do we need to think about age structure? Obviously, COVID-19 was a disease that affected a lot more of our older populations. Many of our resistant bacteria also cause many more infections in our older populations. How does that collision then lead to maybe a sort of skewing more and more resistance infections in the older populations, but fewer in the younger populations? As I mentioned, it seems that kids potentially prescribed fewer narrow spectrum antibiotics in England during 2020. And so analysis of the data and what that data tells us about the trends then tells us what complexity and what structure we need to include in our models. And then the kind of models that I would build would be more on the compartmental ODE side of it to the statistical side. But obviously I would have to do some of those tests and regression analysis to try and understand at a basic level what the key drivers and components of that system should be, or could be, what questions we could ask, and what differences we find in the data.
Thank you very much.
No problem. Any more questions from anyone? I think that it's interesting that 23 of you, or about half of you, thought that COVID-19 will lead to more resistant infections. I think in time for World Antimicrobial Awareness Week, the UK literally released their data last night in terms of AMR infections last year, and so I haven't had time to digest that enough to present to you today, I'm sorry, but that does seem to suggest that there are fewer resistant infections in 2020. So going against what the majority of you thought, and what I initially thought as well, as we talked about briefly then just after the talk, so many people being given antibiotics, but it's a small proportion of the population, and it's a proportion of the population that may be more ill or older anyway. Still needs to be worked through, I think. But I think the other question to do with surveillance is that would we actually know? So would we actually know? Because would we be sampling everybody the same way we were in the first, in a pre-COVID world? So numbers may have gone down, but that's obviously always just reported numbers. So yeah, I need to process that data. But if you're interested, you can look at the UK, it's called the S-Power report, ESPAUR, if you want to have a look.
Yeah, I mean, obviously more research needs to be done on the topic, and everything is very, very novel in that area. But I feel like that just the fact that the preliminary results show that it has increased resistance, whereas your kind of gut feeling would be that it was mine as well. Just like I said earlier, just it makes sense. A disease shows up, we treat it with antibiotics, it, you know, infects most of the world, and you think, okay, and like resistance goes up. It just kind of makes sense intuitively.
Yeah.
And obviously more research needs to be done on the topic, but it just the fact that the preliminary research shows different is quite fascinating. I think it's in its own right.
Yeah, I mean, the other thing to talk about is, I mean, drug-resistant TB. There it seems to be, well, TB in general has massively lost its funding because of COVID-19. So there's going to be big problems there, likely increased incidence, which is just terrifying for a disease that's mostly treatable and curable. But there, you know, from a purely research perspective, it'd be very interesting to see whether drug-resistant TB is transmitting a lot, or whether it is caused by selective pressure infection, which might be selective pressure due to antibiotic usage. But we know that a lot of MDR-TB is due to transmission, and so that is quite worrying in terms of future burden of MDR-TB. Is likely to be higher. But then if we're thinking about something like MRSA, which relies on transmission within hospital settings, it's a very different picture. So yeah, we have to be really careful as well to have the AMR is different in different settings, is different for each bacterium antibiotic. And so the statement of how COVID-19 will affect AMR has to be different, and the answer has to be different depending on what setting you're in. And what is your AMR problem? Like, what are you actually worried about in terms of AMR in your setting? That also varies a lot. People coming in and out of the waiting room. Okay, thank you very much for inviting me. If anybody does have any questions, you're very welcome to send me an email, and I'll try to respond.
Yeah, well, thank you so much for joining us. I personally found this really fascinating. So thank you a lot. And if anybody does have any questions, I feel like they'll be quite free to contact you.
Yeah, of course, definitely.
Thank you so much. And thank you for everybody for joining us. So I like to wish everybody a pleasant day.
Yeah, you too. Bye from London. See you.